Amino acids and proteins. Physical and chemical properties of amino acids and proteins. Classification. The aim of the lesson is familiarize students with the physical and chemical properties of amino acids and proteins. Classification. Objectives of lesson. Describe the structures and functions of the amino acids and their classification. Explain the structures and functions of the proteins and their classification. Amino acids are organic compounds that contain amine, NH2, and carboxyl, COOH, functional groups, along with a side chain, or group, specific to each amino acid. General structure of alpha amino acids. Stereochemistry of amino acids. Configuration for all the common amino acids except glycine. The alpha carbon is bonded to 4 different groups. The alpha carbon atom is thus a chiral center. Chirality: an object that cannot be superimposed on its mirror image is called chiral. The study of fundamental physical and chemical properties of amino acids will help to understand of protein folding, recognition, enzyme catalysis, allosteric regulation. Physical properties of amino acids. Amino acids are colorless, crystalline substance. Most amino acids are tasteless but some are sweet, e.g. glycine, alanine, and some are bitter, for example, arginine. Amino acids have high melting point, 200 to 300 OC due to ionic property. Absorption spectrum. Amino acids absorbs the light at 280 nanometers, so the concentration of amino acids can be measured. Solubility. Solubility of amino acids depends upon polarity, isoelectric point, nature of solvent, pH, and temperature. Amino acids are soluble in water and ethanol, i.e. polar solvent, and insoluble in non-polar solvent like benzene, ether, etc. Amino acids are insoluble at isoelectric point. Solubility depends upon pH of solvent and temperature. For example, tyrosine is soluble in hot water. Titration curve of amino acids. Most of the amino acids are monoamino monocarboxylic acid. When they are in fully protonated form, they can be titrated twice. Titration curve is the graph made between pH of amino acids and volume of acid or base added. It is always sigmoidal. pKa equals tendency of an acid, COOH, to lose proton. This tendency decreases tenfolds as pKa value increase by 1 unit. At pKa, there is equimolar concentration of plus Ve ion and Zwitter ion. Isomerism in amino acids. Except glycine, all amino acids exists in superimpossible mirror image, i.e. D and L form, by Emil Fischer. This configuration indicate absolute concentration but not indicate the optical activity of amino acids. CHO group can be converted into COOH groups, so they are compared. Types of chemical reactions given by amino acids. Reaction due to COOH group, NH2 group, both, R group. Functions of amino acids. Precursor for synthesis of proteins and polypeptides. For synthesis of porphyrin, plant hormones, auxin, lignin, tannin, papaverine, alkaloids, vitamins, ascorbic acid, niacin. The 2 enantiomers of each amino acid are designated by D, L system according to the D and L glyceraldehyde. D is dextrorotation and L is levorotation. Proteins. Physical properties of proteins. Color and taste. Proteins are colorless and usually tasteless. These are homogeneous and crystalline. Shape and size. The proteins range in shape from simple crystalloid spherical structures to long fibrillar structures. Two distinct patterns of shape have been recognized. Globular proteins. These are spherical in shape and occur mainly in plants, especially in seeds and in leaf cells. Fibrillar proteins. These are thread-like or ellipsoidal in shape and occur generally in animal muscles. Amphoteric nature. Like amino acids, the proteins are amphoteric, i.e., they act as acids and alkalis both. The net charge is influenced by the pH value. Each protein has a fixed value of isoelectric point, PL, at which it will move in an electric field. Optical activity. All protein solutions rotate the plane of polarized light to the left, i.e., these are levorotatory. Reactions involving COOH group with alkalis, alcohols, esterification, amines. Reactions involving NH2 group with A. Mineral acids, salt formation. B. Formaldehyde. C. Benzaldehyde, Schiff-S bases are formed. D. Nitrous acid, HNO2, Van Slyke reaction liberate N2 gas and produce hydroxy acids etc. Reactions involving both COO and NH2 group. Reaction with triketohydrogen hydrate, ninhydrin reaction. Reaction with phenyl isothiocyanate or Edman reagent. Reactions involving our group or side chain. A. Biuret test. B. Xanthoproteic test. C. Millen's test. D. Follin's test. E. Sakaguchi test. Solubility. The solubility of proteins is influenced by pH. Solubility is lowest at isoelectric point and increases with increasing acidity or alkalinity. This is because when the protein molecules exist as either cations or anions, repulsive forces between ions are high, and they will be more soluble than in the isoelectric state. Key points. General structure of amino acids. Chiral. D and L forms of AAs. The classification of amino acids. Nonpolar. Uncharged polar. Aromatic. Acidic and basic amino acids. Essential amino acids. Zwitterion, pi. The peptide bond. Glutathione, GSH structure and function. Chemical properties of proteins. Hydrolysis. Proteins are hydrolyzed by a variety of hydrolytic agents. By acidic agents, proteins, upon hydrolysis with conc-HCl, 6,12N. By alkaline agents, proteins may also be hydrolyzed with 2N-NaOH. Thanks for attention.